So the title was the new drugs in the horizon with respect to pancreatic cancer, and what I was thinking of the future in terms of the development. And my talk focused mainly on what is really coming up in the next few years, based on what we have learned from all the years in the past, and based on what is currently ongoing in terms of clinical trials. I focused on several areas which are of interest. One of them was targeting the microenvironment, the stroma. There's a drug which is pegylated PEG-PH20, which is currently in clinical trials, and it has shown promise in select group of patients whose tumors overexpress a protein called hyaluronan, we call it HA. And that's now in phase 3 trials. I also discussed targeting DNA repair pathways that are deranged and abnormal in pancreatic cancer cells, and using them as an opportunity to use drugs such as platinum compounds or even PARP inhibitors. I also discussed other more novel areas of work involving targeting tumor metabolism, and also using drugs that target a protein called mesothelin, and there are a number of approaches trying to use that protein as a target with antibodies or even CAR T cells to try to attack the pancreatic cancer cells. So basically my thought was we learned a lot, and now we're moving into new drugs, new approaches, new strategies, and ultimately, in my opinion, it will be putting everything together, get enough data to generate molecular classifiers of the disease that will help us to really decide what to give to each patient, make it more personalized. We're developing the treatment tools now, where what we're lacking are how do we really apply those treatment tools that we have in treating patients so that we can get the maximum benefit. And in my opinion, it will be combination treatments. But we can't just do combinations empirically. It has to be biologically driven, it has to be rationalized, it has to be personalized, because that's the best way we can not only save toxicity on patients, but also don't do futile treatment that costs a lot but benefit very little. So that's really very important. Just to give you an example, one thing which I discussed also was targeting the stem cells in pancreatic cancer. There is some preliminary data that a drug that targets STAT3 in the cancer stem cells called nab-paclitaxel, when combined with chemotherapy with gemcitabine and nab-paclitaxel, can be producing interesting results. And I think at the end of the day, one would be able maybe to put combinations together. It could be stem cell targeting plus metabolism targeting. It could be stem cell targeting plus DNA repair. But we have to go about figuring out how we can really do these combinations and which combination will really fit a given patient very well. It is a situation where we're moving into big data, because there's no simple data in this disease. Unlike some cancers where there is one driver mutation that you can use one drug and get a very good benefit. Just to give an example, like patients with gastrointestinal stromal tumors, they have c-KIT mutations. You use one drug and you can keep the patient on that single drug for a long time in a very good condition, and patient is in remission. That will not happen in pancreatic cancer based on what we know about it. What will happen is that we have to bring in drugs together. Something else which I talked about in my presentation was immunotherapy. That has really revolutionized how we treat some cancers. In pancreatic cancer, we describe the cancer itself as being an immune desert with the real sense of the word, because there are very few effective T lymphocytes that can kill the cancer cell, but also there are a lot of cells there and other cytokines that produce an immune suppressive environment. And for that reason, we cannot simply give patients what we give, for example, melanoma or non-small cell lung cancer, like PD-1 or PD-L1 inhibitors, and expect a benefit. Vaccines didn't work either. So now there are a lot of combinations that people are testing, which may work to regenerate the T cell function and also try to remove the suppression of the immunity to be effective. In my opinion, the next 5 years will be very exciting, but at the same time, the next 5 years will be will require a lot of hard work, a lot of intense work. And the most important thing is people cooperating together to do this. Because as I said before, we're generating a lot of data, and that data has to be put into some form of a actionable way.